Non-self MHC molecules are recognized by 1 to 10% of T cells. The discovery of MHC restriction by revealing the physiological function of the MHC molecules also helped explain the otherwise puzzling phenomenon of recognition of non-self MHC and the rejection of organs and tissues transplanted between members of the same species. Transplanted organs from donors bearing MHC molecules that differ from those of the recipient, even by as little as one amino acid, are invariably rejected. The rapid and very potent cell-mediated immune response to the transplanted tissue results from the presence in any individual of large numbers of T cells that are specifically reactive to non-self or allogenic MHC molecules. Early studies on T cell responses to allogenic MHC molecules use the mixed lymphocyte reaction. In this reaction, T cells from one individual are mixed with lymphocytes from a second individual. If the T cells of one individual recognize the other individual's MHC molecules as foreign, the T cells will divide and proliferate. The lymphocytes from the second individual are usually prevented from dividing by irradiation or treatment with a cytostatic drug, mitomycin C. Such studies have shown that roughly 1 to 10% of all T cells in an individual will respond to stimulation by cells from another, unrelated member of the same species. This type of T cell response is called alloreactivity because it represents the recognition of allelic polymorphism in allogenic MHC molecules. Before the role of the MHC molecules in antigen presentation was understood, it was a mystery why so many T cells should recognize non-self MHC molecules, as there is no reason the immune system should have evolved a defense against tissue transplants. However, once it was appreciated that T cell receptors have evolved to recognize foreign peptides in combination with polymorphic MHC molecules, alloreactivity became easier to explain. From experiments in which T cells from animals lacking MHC class I and class II molecules have been artificially driven to mature, it has been shown that the ability to recognize MHC molecules is inherent in the genes that encode the T cell receptor, rather than being dependent on selection for MHC recognition during T cell development. The high frequency of alloreactivity of the T cells clearly reflects the commitment of the T cell receptor to the recognition of MHC molecules in general. Mature T cells, however, survive a stringent selection process for the ability to respond to foreign, but not self, peptides bound to self MHC molecules. It is therefore thought that the alloreactivity of mature T cells reflects the cross-reactivity of T cell receptors normally specific for a variety of foreign peptides bound by self MHC molecules. Given a T cell receptor that normally binds a foreign peptide displayed by a self MHC molecule, there are two ways in which it may bind to non-self MHC molecules. In some cases, the peptide bound by the non-self MHC molecule interacts strongly with the T cell receptor, and the T cells bearing this receptor are stimulated to respond. This type of cross-reactive recognition arises because the spectrum of peptides bound by non-self MHC molecules on the transplanted tissues differs from those bound by the host's own MHC, and it is known as peptide-dominant binding. In a second type of cross-reactive recognition, known as MHC-dominant binding, alloreactive T cells respond because of direct binding of the T cell receptor to distinctive features of the non-self MHC molecule. In these cases, the recognition is less dependent on the particular peptide-bound T cell receptor binding to unique features of the non-self MHC molecule generates a strong signal because of the high concentration of the non-self MHC molecule on the surface of the presenting cell. Both these mechanisms may contribute to the high frequency of T cells that can respond to non-self MHC molecules on transplanted tissues. And that's non-self MHC molecules are recognized by 1 to 10% of T cells. Have a great day.